Welcome to another video for biology. In this video we are discussing the transcription of DNA into RNA. Alright, so let's start a discussion. So again, this is about transcription, which occurs inside of the nucleus inside of a cell. Okay, so you have your cytoplasm on the outside. And recall that in the cytoplasm you have free-flowing organelles like your mitochondria, and you also have molecules like glucose floating around. In addition to all of these, and of course there's other organelles that I'm not discussing right now because they're not pertinent to our discussion, but we also have little building blocks called amino acids. Okay, these amino acids are the building blocks of proteins. Proteins are what make up all of our muscles, our hair, fingernails, skin, all the enzymes involved in the various reactions that we've discussed previously. So when I talk about building blocks and proteins, try to imagine walking into a construction site and seeing all the building material kind of just laying around. All this building material needs instructions, and these instructions on where to put what, how to build what, and that goes along with, for the cell, that's our DNA. Okay, our instructions for putting these blocks in the correct pattern are our DNA, which is located in the nucleus. Okay, so again, our DNA located in our nucleus and our building material in the cytoplasm. So there's a bit of a problem because the instructions, again, are here in nucleus and building material on the outside. Okay, and yet our nucleus or our DNA cannot leave the nucleus. It's too large of a molecule to leave. It's in the protective confines wrapped around your chromosomes. Okay, and also our builders, which is going to be our ribosomes. Okay, so you guys remember ribosomes from the study of the cell? Alright, so here's our problem, and this is what we're going to spend the remainder of our video on, is getting our DNA, or getting the instructions that are located in the DNA, the information, the code, the genes, the genes that code for specific proteins that our body needs for various reactions or for building blocks, for building more muscle, for example, after exercise. Okay, so we want to get from our nucleus to our cytoplasm where protein synthesis occurs. Alright, so these are the things that you need to keep in mind as we continue on in our discussion. So DNA is supercoiled in our chromosomes and it won't leave the nucleus, right? And that goes to too large to leave the nucleus. So it transcribes itself into small RNA form, or what we call messenger RNA. Messenger RNA meaning that a message from the nucleus, which is the gene code, a messenger leaves the nucleus into our photosynthesis location, cytoplasm. Messenger RNA, mRNA. So DNA to mRNA. Now, to start off, we want to talk about one of the main characters in this process, and that's RNA polymerase. Okay, that is an enzyme that helps to unzip the DNA molecule, okay, to make a copy of DNA into RNA. All right, so let's take a look at this mRNA molecule in action here. So here I have zoomed in a DNA double helix molecule. Okay, so notice how as this copies, that where a T should go, a thymine should go, you're going to see uracil instead. Okay, so you're going to notice these hydrogen bonds, okay, which are signified by these blue dotted lines. The RNA polymerase is going to break those, okay, and it's going to unwind. Now, at any given moment, RNA polymerase is exposing about 10 to 20 nucleic bases at a time. Here I'm just showing you, for simplicity's sake, one by one. Okay, and it can replace about 60 nucleotides per second. So this is a very, very slow version of the process that I'm giving you here. And again, notice the uracil replacing thymine. Okay, that's a clear indicator that we're talking about RNA, in this case mRNA. Okay, and this process will continue until it is finished. Okay, and as the RNA polymerase continues, the DNA molecule in the back will eventually close up again and create those hydrogen bonds that keep it so stable. Okay, so you're seeing that mRNA, that mRNA, yes, chain get built up one nucleic base at a time. Okay, so we hide our DNA molecule to show our mRNA molecule. Okay, now this mRNA molecule actually goes through a little bit more processing. This is actually called a pre-mRNA. It goes through some processing where some of those nucleic bases actually get snipped off. RNA splicing, it's called. It gets snipped off into what's called interons. Okay, the final product, which is called exons. Okay, so all these are exons. This is a processed mRNA molecule that's ready to leave the nucleus and go into the cell. Okay, let me back up just a little bit actually. Okay, so you'll notice here the mRNA molecule about to go through a nuclear pore. So we're going to zero in, we're going to magnify that nuclear pore, and we're going to take a look as that processed mRNA leaves the nucleus. Okay, so now we're leaving the nucleus and we're going into our cytoplasm where this mRNA molecule will meet up with ribosomes. Okay, and these ribosomes, as we will describe in another video, will go through another process called translation, where we will translate this mRNA code into an actual amino acid chain, a polypeptide, or a protein. Okay, so our message, our instructions, which can now leave the nucleus, will meet up with our builders. Okay, and we will begin the process of protein synthesis. Okay, and that does it for DNA to RNA transcription video. Good luck in your studying.